Theory of Viral Interference, a presentation by Professor Gershom Zaitchik, Faculty of Medicine, Hebrew University of Jerusalem. Baltimore classification of viruses places viruses in 7 classes or groups. And Baltimore classification has 2 interpretations. One says that virus controls cell, since virus is a parasite. And the other interpretation says that cell controls virus and virus is its message. I adhere to the second interpretation and find the virus control cell is false, and all my presentation regard the cell as controlling virus infection, and this is explained by several presentations of me. Now, in order to study the interaction between these viruses, I regard the Baltimore classification as 7 processes triggered by 7 virus classes, and these processes are response to viral infection. So you have the virus interacts with the cell, triggers the formation of provirus, and let the virus also have a messenger RNA. So here you have the standard process of virus interaction with cell. Virus triggers the cell by attaching to its receptor, and it triggers the formation or the reception or reaction of a provirus and messenger RNA, and finally you get a virus following this first trigger. Since several of my previous presentations are represented in this way, I also discuss the process in this reaction between virus and the receptor. So virus attaches to a receptor, triggers the formation of the provirus, and the provirus then controls the formation of virions which exit the cell. So the provirus is a set of cell genes with 3 functions. It stores the self of a cell, it controls who may infect the cell, and controls virus production, and the self is a set of provirus genes unique to each cell. And I describe it in the theory of virus and cell updates. So again, virus attaches to a receptor, triggers the reaction of provirus, and the provirus also controls the formation of virions, and we have the virus exit the cell. Virus is a cell messenger. Virus is created by provirus, and virus stores self of the cell in its genome. And virus infects only cells with the same self, and the self is a set of provirus genes unique to cell. Again, it's explained also in the theory of virus and cell updates. So here's an example of 2 processes. One is class 6 process, and one is class 5 virus attempts to infect a class 6 provirus. What does it mean? Since class 6 provirus controls the cell, this class 5 virus cannot infect the cell, and it will be blocked by the receptor. Again, this class 5 so-called attempt infected cell will continue to produce virions of class 6. We are talking about viral interference, one of the most important discoveries in case of viral infection by Harry Rubin, and he discussed it in several important presentations, and here I cite the early history of tumor virology, Rouse, Riff, and Rav. Here's a reference. So we are talking about 3 viruses: Rouse sarcoma virus, avian leukosis virus, and a resistance factor, which is actually the virus in embryo. So RIF is the ALV virus in embryo, and what Harry Rubin discovered, that the RIF resists RSV infection, which means that first of all you have to infect the embryo, the chicken, with ALV, and then you get this RIF resistance. So here you have an illustration how this is within the set of processes which I just described, because without regarding the classes as classes of processes, it's very difficult to understand what happened during the experiments of Harry Rubin. So we start with ALV, which is a virus of class 6, and ALV is infecting the cell and triggers the formation of provirus of the class 6, and everything as I described before. But we are talking about RSV, which attempts to infect this cell, but the cell is a resistance infection because it was previously infected by ALV. And this is controlled by the provirus, and since this subject is extremely difficult, I shall return to it in future presentation to explain the significance of this viral interference. So, conclusion. Virus is a cell messenger created by the cell. No cell means no virus. And you are invited to listen to more to come, and I hope you enjoy it.